Hello everyone, welcome to this amazing platform of Infinity Learn by Sri Chaitanya. My lovely kids, today we will continue the discussion on transcription process. Remember, in the last class, in the previous one, what we did, we actually took an introduction related to transcription process. What is a transcription process? What is the definition of transcription process? What is a transcription unit that actually participates in a transcription process? All these important information we studied in the previous class. The objective of today's class is to continue this transcription process. So, in this session, we will find out how this transcription process takes place inside a prokaryotic cell, and we will also try to understand some important terms like gene, like cistron, what is a monocistronic gene, what is a polycistronic gene, and also we will try to see what are the various types of RNAs that exist in cells. So, that is the objective of today's session. So, let us start our class. Very warm welcome everybody. Hello Shashank, good evening. Hello Sunil. Let us start the session. And obviously, before starting, I would love to tell you, I request you all to visit infinitylearn.com's website and check out the amazing courses that we offer there. You can use those courses subscription at 40% off by applying the code Kiran40. So, I request you all, do check out. Let us start the class, students. Yes, we will start the class with the very important term, that is gene. See students, what is a gene? Gene is the functional unit of inheritance. That is what your NCERT says. What is inheritance? Inheritance means transfer of characters from one generation to another. So, this whole process of transfer of characters from parental generation to offspring, this process is known as inheritance, and gene is the unit, gene is the functional unit of inheritance. That means genes will help in transfer of characters, right, from parental to progeny, right. Next thing that you need to understand is gene will code for all the three major types of RNAs, tRNA, mRNA and rRNA. It is not like some of you might get confused, or some of you might have this notion that only gene is responsible for producing just mRNA. That is not the case. mRNA, tRNA, rRNA all will be produced from gene, right. So, what is gene? Gene is a functional unit of inheritance. Gene is a segment of DNA from which rRNA, tRNA and mRNA can be produced. Now, let us see what is the another term. Another term is, my kids, it is cistron. What is cistron? Cistron is also segment of DNA that codes for a polypeptide chain. What is a polypeptide chain? Polypeptide chain means a protein. So, what is a cistron? Simple. What is a cistron? Cistron is a segment of DNA, part of DNA from which a protein, a functional protein can be produced or synthesized, right. Now, students, I want to tell you something very important related to the previous lecture. See, remember in the previous lecture, we were discussing about the transcription unit, and if you remember correctly, transcription unit has three main components: promoter, structural gene and terminator. So, the structural gene of the transcription unit from which a polypeptide chain will be produced during translation process, that structural gene can be monocistronic as well as polycistronic. Let us see what does these two terms mean with respect to structural gene. So, structural gene, my kids, can be monocistronic as well as polycistronic. When I say a structural gene, listen this very carefully, the difference between monocistronic and polycistronic structural gene. Whenever I say a structural gene is polycistronic, that means from that particular structural gene, one type of protein can be synthesized, right, and such kind of monocistronic structural gene are most common in eukaryotic cells. What is a polycistronic gene? A polycistronic gene is a type of a structural gene from which more than one type of proteins can be synthesized. So, that is the difference between a monocistronic structural gene and a polycistronic structural genes. Polycistronic structural genes are most commonly seen in bacteria or prokaryotic cell. So, see, let me write it for you. What is a monocistronic structural gene? Monocistronic structural gene is the one from which is the one from which one type of protein can be synthesized. And next one is, they are most commonly seen in mostly in eukaryotes. Please remember this fact. Polycistronic structural gene means, my kids, from which a polycistronic structural gene is the one from which more than one type of protein can be synthesized. Polycistronic, more than one type of protein can be synthesized, and monocistronic means from which one type of protein can be synthesized. They are mostly seen in bacteria or prokaryotes. Please remember this. Let us see what we need to understand next. See students, monocistronic structural gene, as you have studied, as we have discussed on the previous slide, they are most commonly seen in eukaryotic cells. So, in eukaryotic cells or in eukaryotes, these monocistronic structural genes are actually of split type. What they are? Monocistronic structural genes in eukaryotes are split or are of split type. What does that mean? So, see students, split genes or split type of structural genes means these structural genes will be, in them, the coding sequences will be interrupted. I repeat myself, please listen this carefully. Whenever it is said that a structural gene is of split type, for example, which one? Monocistronic. So, monocistronic structural genes are of split type. That means these monocistronic structural genes, coding sequences are interrupted. Who is interrupting these coding sequences? Presence of introns. Presence of introns is interrupting the coding sequences present in these monocistronic structural genes, right? So, whenever inside a gene, inside a structural gene, there are two types of components, exons and introns, then such kind of arrangement of gene is known as split genes, right? So, split genes means there will be presence of introns as well as exons. Now, let us try to understand what is exon and what is introns, right? See here, monocistronic structural genes in eukaryotes are split type because of presence of introns and exons, right? See here, what are exons? Exons are the sequences that actually get coded, right? Exons are the sequences which get coded or expressed in form of a protein, expressed or coded in form of a protein or polypeptide, and what they will appear in a mature or a processed RNA, right? They will appear in a processed RNA, and they should, because they are the one who will be responsible for deciding the sequence of amino acid in a functional protein. So, they should be there. Next, they are interrupted by introns. That means these are the coding sequences. Exons are the coding sequences which are interrupted by the presence of introns inside a monocystronic gene, that is of split type. Right? I hope it will be crystal clear to all of you what is the meaning of split gene when we talk about in text or in context to monocystronic structural gene, right? Next, let us understand what is an intron. Intron are the intervening sequences where they are present in between the exons. Exon, intron, that is the arrangement, exon, intron, and who are they intervening? They are present in between exon, in between intron. So, exon, exon like this. Next, they do not appear in the mature or processed RNA. They should not appear. Why? Because they are not coding for any kind of protein or a polypeptide chain. Hence, they will not appear in a mature or a processed RNA. I hope this much is clear to all of you. Let us see what we need to understand next. See students, let us discuss about the three major types of RNAs that are present inside a cell, right? So, the three major types of RNAs are mRNA, tRNA and rRNA. Please notice what's the name of mRNA. mRNA is messenger RNA, tRNA is a transfer RNA, rRNA is the ribosomal RNA. Please remember these names and not to forget one very important point. All the three types of RNA are important and necessary for the formation of protein. So, all are needed to synthesize a polypeptide chain, that is a protein, right? Let us see what are the functions of these three types of RNAs. Talking about the very first one, messenger RNA. Messenger RNA will actually act as a template, act as a template during translation process. So, the sequence of nucleotides present on an mRNA will decide the sequence of amino acid that will be present in the protein formed from that particular mRNA, right? So, mRNA will act as a template, will decide the sequence of amino acid in a polypeptide chain, right? Next, what is the function of tRNA? tRNA is an adapter molecule. tRNA will actually brings the amino acids during the translation process. So, tRNA will read the codons that are present on mRNA with the help of the anticodon loop and will bring and arrange the respective amino acid in a proper sequence on an mRNA during translation process. Next, what is the function of rRNA? rRNA is the ribosomal RNA. rRNA is the structural RNA. Why it is known as structural RNA? Because it helps in the formation of a structure. What kind of a structure? Ribosome, right? So, rRNA helps in formation of ribosomes. rRNA is also known as catalytic RNA because it helps to catalyze reactions. For example, if I talk about a bacterial cell, in bacterial cells ribosomes are found. What are ribozymes? In bacterial cells ribozymes are found. What are ribozymes? Ribozymes are the one in which 23 srRNA act as a catalyst, right? So, see here, mRNA providing a template, tRNA bringing the amino acid and reads the genetic code on mRNA during translation process. During translation process. Now, see kids, rRNA play a structural role. They helps in formation of ribozymes. They helps in formation of ribosomes. Helps in formation of ribosomes. They act as a catalytic role also. That means they catalyze the reaction. Example, ribozyme. Ribosome, structural. Ribozyme, catalytic. Example, 23S rRNA in bacteria. Right students, one very important information you should note that a single DNA dependent RNA polymerase is required to form all the three types of rRNAs in a bacterial cell or a prokaryote. So, single DNA dependent RNA polymerase. What is a DNA dependent RNA polymerase? It is the main enzyme responsible for polymerizing the ribonucleotide in order to synthesize RNA molecules during transcription process. Right? They will catalyze the transcription of all the types of RNA in a bacteria or a prokaryotic cell. In a bacteria or a prokaryotic cell. I should not write too low. In a prokaryotic cell, that is what I wanted to write. Okay, now let us see, my kids, how transcription process takes place inside a prokaryotic cell. Now the objective will be to understand the process, but before jumping into the process directly, I would love to discuss about this enzyme. Which enzyme? DNA dependent RNA polymerase, which is the main enzyme in the transcription process. So let us discuss in little detail about this enzyme, and then we will directly jump into the process of transcription, how it takes place inside a prokaryotic cell. So see here, first of all, the very first fact that you need to understand is where is this transcription process take place in case of prokaryotic cell, example a bacterial cell. Good evening, NEET aspirant. Wow, what a name. I appreciate that confidence. So, transcription process in prokaryotes, and when we talk about prokaryotes, the transcription process in prokaryotes will occur in cytoplasm. That is the place where transcription process will take place in prokaryotes. Another important thing, there is a single, we have already discussed this fact on the previous slide, a single DNA dependent RNA polymerase will participate in formation of all types of RNAs, T, R and M RNA. Now see students, this enzyme, which enzyme? This DNA dependent, let me change the color for the benefit, for the visible benefit, it will become more visible to all of you. The DNA, no, let me highlight it, that will be better I guess. Highlight it. So we are talking about DNA dependent RNA polymerase, right? So please focus on this highlighted text, right? So see here, what I wanted to explain here is this enzyme DNA dependent RNA polymerase is a complex enzyme. It is made up of 5 subunits. It is a complex enzyme who DNA dependent RNA polymerase, the main enzyme. It is the main enzyme for transcription process. And this enzyme will be made up of this is the entire name of the enzyme. This enzyme is made up of 5 subunits. It is made up of 5 subunits. Right? What are the 5 subunits? The 5 subunits are alpha, 2 alpha units, 1 beta dash and 1 omega. Right? So, 2 alpha units, 1 beta dash unit and 1 omega units are the 5 units making the core part of this enzyme known as DNA dependent RNA polymerase. Right? Why DNA dependent name is there? Because it uses this RNA polymerases uses the DNA template, one of the strand of the DNA to synthesize RNA molecule. So this important, the main enzyme of transcription is a complex enzyme made up of these 5 subunits, and here it is very well represented in this diagram. See, there are 2 alpha units, there is 1 beta dash and omega. So this entire this entire 5 subunit, this entire complex made up of 5 subunits is known as core, core of DNA dependent RNA polymerase. So kids, see this thing. This makes the core of this enzyme, right? There's another unit known as sigma. So the 6th unit, when attaches to this, the 6th unit, the sigma, the initiation factor, when attached to this core of this enzyme, it makes RNA polymerase holoenzyme. This is what? This is the core part, right? The core part of this enzyme. So when with this core part, this is, for example, this is the core part of the RNA polymerase, right? I will directly say now RNA polymerase because saying DNA dependent RNA polymerase is a long name. So from now I will just say RNA polymerase, but you have to understand this is the whole name. So remember, so try to imagine this fist of my hand is what? Core of this RNA polymerase. It is made up of 5 subunits, 2 alpha, beta, beta dash and omega. Now with this core of RNA polymerase, if sigma, 6th subunit sigma attaches to it, which is an initiation factor, now this complex will be known as what? Holoenzyme. Holoenzyme of RNA polymerase, and that's exactly written over here. So these 5 subunits kids, what is this? इस bracket में जो लिखा है वो क्या है? इस bracket में 5 subunits है, alpha 2, beta, beta dash and omega. So these 5 units will make the core enzyme of RNA polymerase. And if this sigma unit, which is the initiation factor, if this sigma, which is the initiation factor, is attached with the core of the RNA polymerase, and if the sigma is attached to it, now this complex is known as holoenzyme, and this holoenzyme is required to initiate or start this transcription process in a specified manner, right? So that's the difference between core enzyme and holoenzyme. I hope this much is clear to all of you. And not to forget, this association of sigma to the core enzyme is actually, this association or this binding of sigma to the core of RNA polymerase is transient. It takes place only for a certain time, just for initiation. So once the initiation of transcription process is completed successfully, perfectly, there won't be any requirement of this sigma factor. Sigma factor will be removed and the remaining core enzyme will do the elongation process. So sigma, association or binding of sigma with the core of RNA polymerase, this association or binding of sigma is transient, is temporarily, only for a certain moment, only for a certain period of time, till the initiation is completed. So the moment initiation of the transcription process is successfully completed, the sigma will breaks off. Sigma subunit will be removed and the remaining 5 subunits of the core of this enzyme RNA polymerase will do the elongation process, right? I hope this much is clear to all of you. Now let us do what? Let us start this process. The process of transcription. Remember, what are we going to do here? We are going to form RNA molecule. Remember my kids, we are studying about transcription process. Transcription process is a process where genetic information is copied from DNA to RNA, and not to forget, entire length of the DNA will not participate, only a segment of DNA will participate in the transcription process, and from that segment of DNA also one of the strand which possess the polarity 3:5 will act as a template strand, right? It will be act as a template strand on which this RNA polymerase will do or catalyze the polymerization of ribonucleotides to form RNA. Please do not forget that. Chaliye. So, the process of transcription will be completed or we will understand in 3 steps: initiation, elongation and termination. What? The entire process of transcription or forming of RNA or copying of genetic information from DNA to RNA will be explained, will be understood in 3 steps. First is initiation, how the process will start. After starting, how elongation will take place. Elongation means binding one ribonucleotide with another, with another, polymerization of the ribonucleotide. And the third one is termination. When this RNA formation of RNA, elongation of RNA is completed, then this process need to be stopped. So, termination of the process. So, under 3 steps we will understand this entire formation of RNA from DNA, transcription process. So, let us start with the very first one, that is initiation. So, in initiation process what will happen? Initiation of transcription process, for this RNA polymerase will bind to the specific sites, specific binding sites that are present in the promoter region. Remember, in the last class we discussed about the transcription unit which possess promoter, structural gene and terminator, right. So, what is the function of the promoter? The promoter possess binding sites or such sequences of nucleotides are present in promoter where RNA polymerase can come and bind and start or initiate the transcription process. So, for initiation of transcription to take place, RNA polymerase will go search its specific binding sites on promoter and will bind there. And this is how initiation will take place. So, RNA polymerase will bind to promoter site and initiates the transcription process. Next, see students, as I told you on the previous slide, for initiation to take place, the core of the RNA polymerase need to be attached with the 6th subunit known as sigma unit, also known as initiation factor. So, when the sigma subunit attaches to the core of RNA polymerase, it makes the activity or function of RNA polymerase more specific. So, because of this binding of sigma factor to the core of RNA polymerase can find its binding sequences on the promoter region. So, this binding of RNA polymerase to the promoter, to the specific site on the promoter, will be catalyzed with the help of this sigma or initiation factor. So, this is catalyzed by sigma factor or initiation factor. What is catalyzed? Initiation of the transcription process is catalyzed by sigma factor, or binding of the RNA polymerase to the specific binding sites on the promoter region. This is catalyzed with the help of sigma factor. Sigma factor will tell RNA polymerase to go and bind on this particular binding site of RNA of promoter region, and from there it can easily start the transcription process. So, I hope you are understanding how sigma factor is providing specificity to this RNA polymerase. It is actually telling the RNA polymerase to go and bind there. It is the place from where you should bind and start the transcription process. I hope I made this clear to all of you. Hello. Yes, I don't know. Yes, I do need that, Jatin. Thank you for letting me know that. And thanks for the appreciation as well. Next, sigma factors confers the specificity to RNA polymerase. I already explained this point. Because of the binding of sigma factor to the RNA polymerase know exactly where to go and bind on the promoter region and start the transcription process. Next, in absence of sigma factor, RNA polymerase will not have the specificity. RNA polymerase won't know where exactly I should go and bind so that I can start the transcription process. So, in the absence of sigma factor, RNA polymerase will start the transcription process from any place. So, in absence of sigma factor, transcription starts non-specifically by core enzyme. So, core enzyme is capable to start, but it won't be specific. From where it was supposed to start, it won't start. Please note this point. So, here you can see, kids, see initiation, elongation and termination. So, we actually discussed about this step, right. So, see what is this? This orange portion is the RNA polymerase, and this is the sigma factor, RNA polymerase core enzyme. This is RNA polymerase core enzyme, and association of RNA polymerase core plus sigma factor will actually helps in or catalyzing the initiation of transcription process. And what is this? This is the promoter region. So, if RNA polymerase, see kids, RNA polymerase, core of RNA polymerase with the help of sigma factor has identified its place in the promoter region, and finally it has bounded itself, right. RNA polymerase has bound itself to this segment of the DNA or to this portion of the transcription unit. Why? Because this is the promoter region which has binding site for RNA polymerase, and this is how the initiation will take place. Now, let us see what happens during the elongation process. If you notice that in the elongation process, what is happening? The sigma factor is actually removed, right. During elongation process, the sigma factor is removed because this core enzyme, this core of RNA polymerase, the core of the RNA polymerase is actually capable enough to do the elongation, to do the polymerization of ribonucleotide alone. So, in elongation process, there is no requirement of sigma because core enzyme of RNA polymerase is itself capable to execute that, right. So, sigma is required only in initiation because it will provide specificity to the RNA polymerase. That means कहाँ पे जाके attach होना है ये बताएगा सिग्मा, किसको? कोर एंजाइम को ऑफ आरएनए पॉलीमरेज। चलिए आगे देखते हैं स्टूडेंट्स, थोड़ा सा और डिटेल में देखते हैं इलोंगेशन प्रोसेस को। Okay, so I wanted to write here elongation. So instead of that, initiation came here. So what is this? This is elongation. Oh no, it's right, it's right. I wanted to explain some more points about initiation over here. So before jumping into the elongation process, there are few more important facts that I wanted to explain about initiation. See here students, please notice the very first one. During the initiation process, what will happen? See, RNA polymerase also uses nucleoside triphosphate as substrate. Yes, ribonucleotides will be used as a substrate to polymerize, right, to form an RNA molecule. Another, polymerize. Who will polymerize? RNA polymerase will polymerize in a template dependent fashion. What does that mean? This means this RNA polymerase will use one of the strand of the segment of the DNA to form a sequence or to form an RNA molecules. So the sequence of nucleotide present on the template strand of the DNA will actually decides the sequence of nucleotides present on RNA molecule. Hence, this RNA polymerase will do what? Template dependent activity. Next, while doing the activity, RNA polymerase will do what? It will follow the complementary rule. Who? Actually students, what are we doing? I am on this slide trying to explain you some of the important properties and function of RNA polymerase. So before jumping into the elongation process, which will be executed by core enzyme of RNA polymerase, it is important to understand what will be the role and function of the RNA polymerase. Second, third, the RNA polymerase will follow the complementary rule, that means opposite to A, right? That means opposite to A, who will come? U, and opposite to G, who will come? C. So, in place of thymine, talking about DNA, DNA has thymine. So, in place of thymine, RNA will possess uracil. Not to forget, RNA will be single stranded. Next, RNA polymerase will also facilitates the opening of the helix. So, remember students, if RNA molecule needs to be synthesized using DNA, one of the DNA as a template strand, so the DNA helix needs to open, right? Until and unless the helix is opened, RNA molecule cannot be synthesized or transcription process cannot takes place, right? So, note this point, when I was explaining you replication process, who was helping there? DNA. In that DNA replication process, helicase enzyme was responsible to open the helix. But unlike the replication process, in transcription process, it will be who? It is RNA polymerase itself. RNA polymerase itself will do the opening as well. So, RNA polymerase is responsible for elongation. RNA polymerase is also responsible for opening the helix for the transcription process. See here. Next, let us now start discussing about the elongation process. So, elongation process, as I already told you, will be catalyzed by the core part of the RNA polymerase. So, catalyzed by the core enzyme of RNA polymerase. So, during elongation process, from this core, the sixth subunit sigma or initiation factor will be removed, and the alone core enzyme that is core of RNA polymerase will do what? Elongation. That means catalyzing the polymerization reaction, polymerization of ribonucleotides to form RNA. Only a short stretch of RNA remain bound to the enzyme. Very simple. This is NCERT line, and you can find that only a small stretch. See, this is the entire length of the RNA that has been produced till now, and from that only a small stretch, only a small segment of RNA will be remain attached to the enzyme during elongation process. Now, after the entire length of the RNA is produced, what will happen? The last step will be termination. Termination of the process, that means stop of transcription process. Again there will be binding of another factor to the core enzyme, to the core RNA polymerase. This time who is going to bind? This time termination factor, that is Rho factor, will bind to the core enzyme of RNA polymerase. So, if this is the core enzyme, this is the core of RNA polymerase which was till now doing elongation process, catalyzing the ribonucleotide or polymerizing the ribonucleotides, right. So, this core enzyme of RNA polymerase, from this to this core enzyme, another factor, another unit will bind again transiently, temporarily, for a short time. Who? Rho factor. So, Rho factor will bind to the core of RNA polymerase and will stop the transcription process. When the transcription process will be done, or when this process of transcription will stop? When this RNA is completely synthesized. Whatever the sequence of nucleotide it was supposed to possess, it has done, it now possess. Whatever RNA was supposed to be synthesized, it is now completely done. From this point, core to the core of RNA polymerase, Rho factor will attach and stop this process. So, for termination to take place, another factor known as termination factor. So, this is Rho factor, which is also known as termination factor, also known as termination factor. So, this rho factor or terminator factor is required to terminate or stop this transcription process, and during termination what will happen? This nascent RNA, what is a nascent RNA? Abhi jo bana hai, turant freshly prepared RNA. So, this nascent RNA, right, and RNA polymerase will fall off. This nascent RNA and RNA polymerase will fall off, will get detached from this strand of DNA or from this segment of DNA that was taking part in transcription process. This is how termination or stop of transcription process will take place, right students? Now, there is an interesting question that is given in your NCERT. Let us discuss about that. So, this question says how RNA polymerase is able to catalyze all the three steps: initiation, termination as well as elongation. So, initiation, elongation, termination, how come all are catalyzed, or how come RNA polymerase is able to catalyze all three? So, see student, it is not the case. RNA polymerase core enzyme of RNA polymerase can only do elongation. It is association of sigma factor and rho factor that provides specificity to this core of RNA polymerase, with the help of which RNA polymerase can initiate and terminate the transcription process. So, I repeat myself students, it is not the case that RNA polymerase, it is not the case that alone the core of RNA polymerase cannot catalyze all three. Core of RNA polymerase can only catalyze the elongation process, right, can catalyze the polymerization of ribonucleotide. When this core of RNA polymerase get or binds with sigma factor, it can do initiation. When the core of RNA polymerase binds to the termination factor or rho factor, it can terminate or stop the transcription process. So, RNA polymerase is only capable of catalyzing the process of elongation. What? RNA polymerase core enzyme. RNA polymerase, maybe we are talking about the core enzyme, right? It associate transiently. Transiently means, better transient means temporarily. So, temporarily it will associate with sigma factor, which is the initiation factor, and with rho factor, which is the termination factor, to initiate and terminate transcription respectively, right? Next, association with these factors alters means change, alters or change the specificity of RNA polymerase to either initiate or terminate. So, this RNA polymerase will do initiation or will do termination. That will be decided by the factor who is binding with it for the short duration, for temporary time period, for transient, right? So, see this RNA polymerase will do initiation when it binds with sigma, this RNA polymerase will do termination when it binds with rho factor. So, this is how the specificity got altered of RNA polymerase when it binds to sigma and rho factors. Last thing that I need to discuss in this session is students, in bacteria many a times, in bacteria means in prokaryotes, right? So, in bacteria or in prokaryotes many a time translation, what is translation? Formation of protein. What is translation? A process in which there is formation of proteins. Formation of proteins. So, in bacteria many a times translation, that means formation of proteins can begin much before the mRNA is fully transcribed. So, before the formation of a mature mRNA, or I can say before an mRNA is completely synthesized during transcription process, many times in bacteria or prokaryotic cell, translation process also begins. So, in prokaryotes and bacteria, this transcription and translation is a coupled reaction, can take place simultaneously, right? Why? Because of the two reasons. Why? Because of the two reasons. The very first reason is mRNA that is synthesized in prokaryotes do not require any processing. It can directly participate in translation, right? Unlike that in eukaryotic cells, first of all, a primary transcript hnRNA is produced, and after processing hnRNA get converted to mRNA, which actually participate in translation process. But such scene is not there in a prokaryotic cell like bacteria. Here the nascently formed, freshly formed mRNA without processing directly participate in translation process, formation of protein. And second, in bacterial cell, the compartment or the place where transcription and translation will take place is same, because they do not possess what? Well defined or cell or membrane bound cell organelles are absent in bacterial or prokaryotic cells. Hence, the place where transcription and translation will take place is same. So, these are the two important reasons because of which many a times in bacteria transcription and translation are coupled processes, right. mRNA does not require processing, and transcription and translation takes place in the same compartment. These are the two important reasons. So, that is all students I wanted to explain in today's session. Tell me, is there any doubt till now? Any doubt till now, my kids? Good evening. Thank you. Thank you, simple girl. You are new girl to this channel. I welcome you, and do not forget to join the next session. Okay. So, thank you student. That's all from today's session. Please give me a thumbs up if you like me and this session, and do not forget to subscribe Infinity Learn channel by Sri Chaitanya. Also hit the bell icon so that you will be notified every time whenever I will go live, and you will not miss any single session in future. And also do not forget to give me a comment. Thank you students. That's all.